Okay, let's get started. Uh, Okay, so hello everyone. Welcome to week 10 of NGS data analysis and applications. So in the last weeks, we learned about RNA sequencing data and how we pre-process the data, how we align the data, then we do differential expression analysis with different types of models, and ultimately functional enrichment analysis. So this class will be mostly covering on the hands-on aspects of RNA with RNA-seq data and how we do that, and like mostly we'll like do it through R. So like today is a hands-on session, you can say, and yeah, we'll go through it the whole thing, starting from counts data and how we normalize and stuff to functional enrichment analysis. So okay, let's get started. Okay, yeah. So first, let me give you the overview of the data, what it looks like and everything. So this is the data. This is the GOID of the GAC ID of the data, which is basically is yeah. Okay, yeah. So this is the data set that we'll be using for today's analysis. So basically, this is the paper associated with it. So this is an RNA sequencing data, which is uh, this experiment is performed on mouse, and basically it is a transcriptomic analysis of luminal basal cell subpopulations in the lactating versus pregnant mammary gland of mice, and they have done RNA sequencing. So we have 12 samples here. So each of the sample information is given within this GSM IDs. So this is a luminal virgin sample of the mammary gland. So it's a wild type sample. It's a mouse mammary gland tissue, and it's in the virgin developmental stage. So for all the information that you need required for each of the samples, it is given here. So for example, this one is a virgin basal cell population, and the others are, say for example, this is the lactating day two lactation developmental stage of the luminal cell population. So all these samples are taken from mouse models, and they are uh, they've undergone RNA sequencing, and what we want to see is basically how different genes are expressed in different types of condition under different types of conditions for this particular uh, data set. Yeah, so this is the paper if you want to take a look. So I'll just quickly move to the methods here. So yeah, so these are your mice models here, and uh, they have given some treatment for the mice for different time points, and all of them is described here. So we'll not go into the details of this, but let's start analyzing the data. Okay. Yeah. So this is the whole data set. Now, if you want to get the counts matrix, you use the FTP server or directly downlink from the HTTP server here. This is a .gz file you need to decompress, and yeah, yeah. So the file that is downloaded is a gz file. If you decompress, it'll give you the matrix files. Yeah. So this is the data set that we have for today. So these are your Entrose gene IDs. So these are basically Entrose IDs which directly map to particular genes. These IDs and the genes are mapped. These are the length of the IDs, as in the length of the genes which we can use to normalize, and these are your different samples. So these are the sample names, and each of the each of the value represents the counts of that particular sample for that particular gene. So the gene is given by these IDs, and the samples are given by your column headers. So this is how the data set looks like. So we have in total around 27,000 genes here. We'll not use all of them. We'll remove some of them during QC and stuff, because yeah, during pre-processing we'll remove certain of these samples and values. Not samples, the genes that are not particularly well expressed here. So for example, we have certain genes which are all, like, so for example, this one, this is not expressed across any of the tissue, any of the samples here. So we'll not use those. We'll remove this during the QC stage. But yeah, this is how the data set looks like, and each of the samples are corresponding information is given in this particular GSM, under this GSM IDs for the details of the samples. You can take a look here at what day of pregnancy it is, what is the immunophenotype, and so on. Yeah. Okay. So this is the data set that we are going to use, and let's get on. So what we'll do is basically first we'll do some pre-processing, then we'll do differential expression analysis, and then we'll move on to functional enrichment kinds. Okay. So these are the mostly uh, the libraries that we are going to use. Along with this, we are also going to use the DESeq2 library. But yeah, in the later stages, I'll add that. Okay. So we have ejar, lima, gplots, and this is basically a database which gives you the mouse model, basically the mappings from Entres IDs to your gene names. So yeah, okay, yeah. So we have loaded all these packages. So first of all, after loading, we need to set the working directory. So you can either do it like this: you go to the session tab and you go to set working directory and choose directory. So my all my data is here in download, so I'll just put here only. And if you don't do it like this, you can do it using set wd. So it works both ways. Okay, so yeah, now we have set our working directory and everything. Now let's get the data. So if you download the counts data, so this is the name of the counts data. So let's get it. This is your seek data. So if you look at your seek data, it's this data set only that has been loaded here. So you do a read.dlim and you, sorry, and you give the name of the file, and basically it will read the data here. And if you see, this is loaded as a data frame. Okay, so yeah, let's move on now. Now we don't want, we just want to pre-process the data for now. So let's just remove the first two columns and store them as the basically the IDs of the genes and everything. So we remove the first two columns. Then we store the Entres gene IDs as your row names. So now if you see your counts data, this is your counts data. This is how the data set looks like. So these are your row names, which are your gene IDs. These are your Entres gene IDs, and these are your different samples. And these represent the number of counts for that particular gene for in that particular sample, right? So it's pretty straightforward. Okay. Okay, let's do it here actually. Yeah, and we also have a sample info file. So this is the file that is looking here. Uh, yeah, so this is a sample info file. So basically, which is the sample ID and the sample name and the cell type and the status of the lactation is given here. So this is a file that we make custom made, but in some cases you'll have sample information files given in the INSEQ dataset also. Okay, so okay, let's move on. Okay, so let's read the sample info file as well. And if you see the view sample info, this is how the sample info looks like. Now let's lead the sequence data. This is the head of the sequence data, as I was saying. So this is your length, your interest gene IDs, and your rest of the samples here. And let's check the sequence. So this is the view one. So let's check the basically the dimension of the data. So we have 27,179 rows and 14 columns. So what it means is we have 27,000 genes and 14 samples here, right? So this is the one. So the seq data object contains information about genes, which is less one gene per row. The first column has the entries gene ID, as I was saying. The second has the gene length, and the remaining columns contain information about the number of reads aligning to each gene in the experimented sample. Just a second. Yeah. Hmm. Yeah. As I was saying, uh, one gene per row. The first column has your gene ID. In some cases, you will not have the gene ID. You will directly have the gene name. So, but in this case, we do not have the gene IDs. We need to map the gene IDs to the gene name in the further steps. But other than that, you have the gene IDs. The second is the gene length, and the remaining column contains information about the number of reads aligning to each gene for that particular experimental sample. There are two replicates for each cell type and time point. So all these details can be found in the series matrix file. So I'll just quickly show you where the series matrix file is. This was the geo page. So here, sorry, this is the GSM ID. So yeah, this is the original geo page. This is the GSA ID here, and here is the series matrix file. You can just click on it. Here it will take you to the FTP server, and you can download this here. And if you unzip it, this tells you all the information for the particular for this particular sample. So for example, this is the first sample titles for all the 14 samples here. You have the GSM IDs associated with these 14 samples. Uh, all this information, summation date, update, organisms, which organism, all are mouse samples. So all are muscular samples, so on. And you have other information as well, background strain. So this is also given here, genotypes and everything, gender is also given here. So all this information you can find in the series matrix file. So yeah, a lot of information is present here. So if you want to have further details about the samples, you can go and look into the series matrix file. Okay. Now, uh, there are two replicates for each cell type and time point. So yeah, and yeah, this is in the geo page, this idea. So I'll just write the ID in the chat box if you want to analyze this in your time. So you can just go to GEO, gene expression omnibus, and you can just search for this ID and you'll get the data set there. Okay. Now we will be manipulating and reformatting the counts matrix to a suitable format for downstream analysis. The first two columns in the seq data frame containing the annotation information. We need a new matrix containing only the counts, but we can store the gene identifiers as row names. We will add more annotation information about each gene later in the slides. Okay. So we're creating a new object called count data that will only contain the counts for these 12 samples. Okay. So we're removing the first two columns from here. So okay, and this is the count data. We have removed all the first two samples from it. Okay. Now we set the row names as the gene IDs. So now counts data looks like this. Your row names have the gene IDs. Okay. I'll not go here. If you look at the columns of the counts data, this has all the 12 sample IDs as your columns. Right? Uh, now these sample IDs are like pretty long, so we'll shorten them, shorten these to contain only the relevant information about each sample, and we'll use this substring command to extract the first 7 characters and use this as column name. So this is the command for that. Sub str is the command. So we use that and we convert the column names to shorter names like this. So see the column names, these were the initial column names. Now this has been shortened to this. We only like the first 5 or 6 characters we are keeping here. Now that this is done, uh, now the column names are now the same as the sample name and the sample info file. This is good because it means our sample information in the sample info is in the same order as the columns in the count data. Now, if you check the number of columns here, it's basically 12, which is the number of samples we have. Now, the next step is removing the lowly expressed genes. So, genes with very low counts across libraries provide little evidence for differential expression, and they interfere with some of the statistical approximations that are later used in the pipeline. They also add to multiple testing burden when estimating false discovery rate, reducing power to detect differentially expressed genes. These genes should be filtered out prior to analysis. And there are a few ways to filter out these lowly expressed genes when there are biological replicates in each group. In this case, we have a sample size of 2 for each group. So for one condition, we have 2 replicates here. So we favor filtering on a minimum counts per million threshold present at least 2 samples. 2 represents the smallest sample size for each group in our example. In this dataset, we choose to retain genes if they are expressed at a CPM, which is the counts per million, above zero point 0.5 in at least 2 of the samples. So we how do we get the CPM values, counts per million values? We'll use the CPM function from the HR library to generate the CPM values and then filter. Note that by converting to counts per million, we are normalizing for different sequencing depths for each sample, right? So we'll just convert it into the counts per million data. So if you just convert it, so this tells you which are the counts per million data. Now if you go ahead here, so see all of them have been converted to counts per million. Now you see these are the ones which we do not want. So this expression, this particular gene is not expressed across all the 12 samples here. So these are the kinds of things that we need to filter out. These are the lowly expressed genes here. So we take a threshold of zero point 0.5 and we filter out the genes that have basically a CPM value of at least zero point 0.5 and above. So we do this thresholding now. This is basically the binary Boolean representation for that, which is like true or false for if it is above zero point 0.5 or if it is below zero point 0.5. So our condition is it has to be above zero point 0.5 in at least 2 of the samples. Then we'll remove them. So this gives the total number of basically how many trues are there in each row, meaning how many samples have a CPM greater than zero point 0.5, and there are total uh, 11,433 genes that have true in all 12 samples. So we need to remove those. We would like to keep the genes that have at least 2 trues in uh, each of the rows of the threshold. So we are keeping at least 2 samples which have CPM greater than zero point 0.5. So this is the command for that, keep, which is the row sums of threshold greater than equal to 2. If we do that, now if subset the rows of the count data to keep the most highly expressed genes. So this is the subsetting. Now if you do the summary, it gives you the, now you see the number of genes that we have come to. So initially we started off with how many? Uh, we started off with 27,000 and now we are down to 15,000. So a cutoff of zero point 0.5 has been taken, and like we are assuming like the counts per million value should be greater than equal to zero point 0.5 in at least 2 of the samples here. So if we do apply that condition, we are down from 27,000 to 15,000 genes. These are the more expressed genes, and we have removed all the lowly expressed genes so that they do not interfere with the statistical analysis. Counts per million of zero point 0.5 is used as it corresponds to a count of 10 to 15 for the library sizes in this data set. If any count is smaller, then it is considered to be very low, indicating that the associated gene is not expressed in this cell. A requirement for expression in the two or more libraries is used as each group contains two replicates. This ensures that a gene will be retained if it is only expressed in one group. Smaller counts per million thresholds don't usually appropriate for large libraries. As a general rule, a good threshold can be chosen by identifying the counts per million that correspond to a count of 10, which in this case is about of zero point 0.5. You can filter with the counts per million values rather than filter on the counts directly, as the latter does not account for differences in library sizes because between samples. Okay. Uh, there can be other metrics that you can use. For this one, we are using counts per million. So you can use RPK, you can use FPK, you can use TMM, and other metrics you can use. So there is no like restriction on that. So for this our case, we are using uh, counts per million. That's all. Okay. So now that we have filtered the data, so just quickly. So this is our filtered data. So we have around 15,804 genes and 12 samples that we already had. Now, uh, we use CP counts per million just to filter the data. The counts data that we have right now, it's just the counts data. We have not normalized with anything to counts per million or RPKM or FPKM or anything. So this is just denoting the counts, but we used counts per million to filter out the lowly expressed genes here. Now, if you check the plot, it's kind of good. So if these are your counts per million values and these are your counts data, so you have certain genes which are like having high counts and high count data in the sense of uh, basically your counts per million value is also high, your counts value is equally higher. So this is a good uh, way to see if your genes are well expressed across the sample. So yeah, this is just a preliminary thing to do, and look at you can look at the you can limit the x and y axis so that we can look at what are happening to the smaller counts as well. So you use the plot function and you just put x limit and y limit here. So this looks like this. So these are on the smaller side of your counts per million, and it's actually very well correlated here. So this works fine. Okay. Okay.
Hi, can you zoom it out a bit because the letters are very small.
Sorry.
Can you zoom it out the screen? Yours,
I don't know how to do that.
yeah,
One
because
second.
the letters, the console is very small to see.
Oh, the console you don't need to bother about. The code is written here only in your source page. But it's if I execute that code, it'll come in the console uh, here. But actually, the code is written here. So I'll try to zoom out. If this works. Now, is this better?
Okay, to some extent. Yeah,
Yeah, lower than this, I cannot see. Yeah, good.
yeah, yeah. Fine, fine.
Okay. So yeah, we're just zooming in into the lower thresholds, lower counts per million, and it's actually very showing very well. So our data is okay in that way. So now, what we want to do is we want to get the DG object, the differential gene expression object. Will create this list object, and this object can be used by HR to store the counts data. It has a number of slots for containing various parameters about the data. So an R object is it's a collection of certain types of data, and it can not only have one type of data, it can have different types of data associated with a particular thing. So for example, this in this one will have many information about how are what are the differentially expressed genes, what are the FDRs, and other information about the samples and everything. We can store it in an object. Like we can have different data types and we can put those together in an object. The DG object, the differential gene expression of object, is something like that. Okay, so this is this function. Let's show you. I'll show you here. So this is a lemma. This object is a lemma object. So basically, this is its usage. You have the counts matrix. You can give the library size, or which is basically the column sum of the counts. Sorry, yeah. You can give normalizing factors, and if not, you can just give the counts matrix. And what it will give you is basically something like this. So it will give you. Uh, See, I'll show it in my example. So this is your, we are just giving the counts which we have uh, QC'd. We are just giving the counts as the input and DG list. So we are making the object. So this is the DG object that has been formed. So if you see on the right here, this is the DG object here. So see, you have the counts information here. You have the sample information here. Right. So like this is an object that has multiple data types incorporated in that. So yeah, and this is part of the Lima package. So you need the Lima package to run this. So if you're facing at any point of time, you cannot like the package is not installed or anything. So either you can directly install it from here. So basically you go to tools and install packages from here. You can write the name of the package and click on install, it will install. But for the bioconductor packages, the specific for biological data analysis, some of the packages are in bioconductor. So they are not in the normal CRAN repository. So you need to go to bioconductor here. Yeah. Say for example, you want to just a second. Yeah, you want to like install say lima install. You go to this website and you'll have to first install bioconductor. So it checks if you have bioconductor or not. You have to first install bioconductor, and then through bioconductor BioC manager you need to install these packages. So most of the packages that we'll use, including HR, lima, DSIC2, these kind of packages, these are all part of bioconductor. These are not present in your CRAN repository. So you cannot directly install it from this like tools and install packages from here. It will not come. So you have to go through bioconductor. Okay. So yeah, coming back. So this is the DG object that you have, counts data, and with all the sample information. Now, if you do what slot, if you want to check what slots are stored in the DG object, you do names, and it has two uh, slots. One is for counts, one is for samples, as I showed. And if you just want to check what are the samples, you do a DG object, and you call the samples basically sample slot here, and it will give you what are the samples. So see, we have not defined any groups or anything. So by default, it takes group one for all of them. So these are all your sample IDs and your library sizes that were already present in the sample information column. And these group and normalization factors, we have not done anything with it. So it basically takes it on its own. So now we have done the DG object creation. Now we want to do quality control. Now that we have got rid of lowly expressed genes and we have counts in our DG list object, we can look into a few different plots to check that the data quality is good. and the samples are as we would expect. So first we need to check how many reads we have for each sample in the DG object. So if you want to check the library size, so basically you want to see this column basically. Yeah, now we want to see the library sizes. So if you just do this, these are all your library sizes that we are getting from here, right? Now, yeah, you want to say, for example, I think we don't need to see the distribution of the library sizes because we'll not go into the details of the library sizes. It was just the information that was there I wanted to show. Now we want to get, say, for example, uh, log transformation along with counts per million for the counts data. So counts data will not be normally distributed. So if you want to examine distributions of the raw counts, we need to take log transformation for the counts. Next, we'll use box plots to check the distribution of the read counts on the log2 scale. We can use the CPM function to get the log2 counts per million, which are corrected for different library sizes. The counts per million function also adds a small offset to avoid taking log of zero. So if you, for example, have zero in your counts, it'll add a very small amount so that when you're taking log, it does not become an issue and give you an error. So you take log2 counts per million of the object that we have, and you say log equal to true. This becomes the log count. So if you check the log counts here, so see your basically first it has been transformed to counts per million, then it has been log2 transformed. So this is how your data looks like. Now this will be normally distributed. If you see the want to see the box plot, it's it's actually pretty good. So the log value has scaled your the number of counts that is there, and basically all of them are around the similar amount. So it is not like the scaling is not an issue here. So we have along all the samples, we have the similar kind of scaling for the counts matrix. Okay, what we want to see is multi-dimensional scaling plots, which basically uh, will tell you one of them is using PCA or any other technique as well. So this will tell you which are the samples that are like having similar kind of expression, which are the samples that are not having similar kind of expression. So by far one of the most important plots that we make when we analyze RNA-seq data are MDS plots, which has the multi-dimensional plots. An MDS plot is a visualization of principal component analysis. So you do PCA and you kind of try to cluster the samples together, which determines the greatest sources of variation in data. A principal component analysis is an example of unsupervised analysis. Excuse me. Where we don't need to specify the groups. If your experiment is well controlled and has worked well, what we hope to see is the greatest source of variation in the data are the treatment groups that we are interested in. It is also incredibly useful tool for quality control and checking for outliers, and we'll use this particular uh, function plot MDS to create this MDS plot. So you do a simple plot MDS with the DG object, and this is what you get. So basically you see these are the samples that are clustering together, these samples are clustering together here, here, and here. So basically we are doing kind of PC1 versus PC2 here. Now what is happening is, if you have batch effect in your data, say for example you have variation in your data that is arise due to technicalities of the sequencing and not due to differences in gene expression, what will happen is similar type of samples like two replicates you have for each condition, right? Now for each condition these two replicates should cluster together. For example, here these are the two replicates, here are the two replicates, so these are clustering together here. But if you had batch effect in your data, what will happen is these two replicates will not cluster together, this will cluster separately. So that is a very good indication that that your data has batch effect and you need to remove it using some sort of model. But in our case we do not have batch effect here. So these are just the sample IDs. In the later ones will like include the sample information here so that we can have a better understanding is what samples are clustering together and what samples are not. right? Now, yeah, it is a bit difficult to see exactly what is going on with the default plot, although we do see samples grouping together in pairs. To make this plot more informative, we can color the samples according to the grouping information, and we can change the labels or instead of the labels that we have as points. That is what we are exactly going to do. So if we do this, and basically we are specifying the option to let two plots side by side, and first is this is the command for that part, and basically we want to see how many cell types and in what order are they stored. So we do a levels for that, and we choose different colors for different samples, types of groups, and then we put them in a data frame. Now you do the MDS plot again, right? So this we need to add the legend. So let's add the legend here. Uh, just let me check this one. It is not coming here. Basically, mm, yeah, I think this should work. I'm not sure why this is not working. Actually, I have done this previously once. Let me show you that. Yeah. So basically, here we are grouping that. So uh, yeah, from the sample info. So you have the sample info here, and then you group it, and this is the group. So basically you have these set of 12 groups. So we have basal virgin, two are basal virgin, two are basal pregnant, two are basal lactate, two are luminal virgin, like that. And now if you plot that group here, so this is your design matrix. We'll come to the design matrix later, but I just want to show you the MDS plot. Yeah. Now, if you just put the groups together and basically and plot the MDS plot, instead of just having the IDs, you put the group labels on that. You see all the like similar type of uh, samples are clustering together. So luminal virgin, these are the two samples here that is clustering together. Luminal pregnant, these are the two samples here, they are clustering together. Luminal lactate, two replicates. Basal virgin, basal pregnant, and basal lactate. So the similar type of conditions that you have for uh, each sample. So if the samples have similar conditions, they're clustering together. So that this means your data does not have batch effect. The variation that you're seeing, why is basal virgin so separately clustering from luminal virgin? Because the gene expression is different. It's not because you have batch effect in your data. So this is what we were trying to do. Now if you want to estimate the dispersion in the data, like how spread out your data is, you do estimate common dispersion. This is basically, I'll show you the R page for that. Yeah, so this takes the matrix counts of a DG object, and yeah, it's like this. So you have the matrix counts, you give the counts, and you give the groups, and it measures the like how spread out your data is. Right now, if you go here, so yeah, this is your common dispersion. So basically, what this plot is telling you is your biological coefficients of variation along with your CPM, average CPM. So this is the blue line is showing you how your dispersion is changing across your if you if across your counts per million, and how it is actually would have changed if there were no variation in your data and your samples were similar, having similar type of gene expression. Now, it is not the case here. You have quite an amount of variation, which is getting showed by this dispersion line, which is the blue line here for this one. Right now, if you want to estimate other types of dispersion, so this was the common dispersion technique. Now, if you want to estimate gene-wise dispersion estimates, you use GLM trend dispersion. So GLM is the generalized linear model that you use to estimate the dispersion. So this is your one dispersion. This is your other one. Now, if you want to plot the estimated dispersion, is the BCV. So basically, biological coefficient of variation, what it is defining. And if you plot it, this is what it comes. So here also you see the um, the the dispersion line is basically not the same for all the groups. So basically, if you have different groups of data, it is the dispersion is showing that across your samples, there is quite amount of variation in the data compared to a control sample that you'll have. So the red line is telling you the control sample, and basically your blue line is telling you how your current set of samples are well dispersed. So you have a lot of variation in your data. That is what it is trying to do. Now, what we'll do is we'll try to test for differential expression. Now we have done some QC. We took the data, we converted to CPM, then we trans. We had the log transformation. Now, just a second, guys. Yeah, sorry. Okay. Uh, yeah. Now we next we fit is the gene-wise GLMs for differential expression analysis. Now, this is your function for that GLM fit. This also comes with the lemma package, and basically we are going to fit the model for differential expression analysis using the DGE object. Now we use the fit. So this is the if you fit it, you get this output. So for each gene, so these are the first 10 rows here. So for each gene, you have a log fold change, where at the log counts per million values, which is the expression value there. Then we are here looking for the p-value, and we have not adjusted the p-value for multiple hypothesis correction yet, but these are your FDR values, which are your false discovery rates. So if you fit a GLM, you get along with your basically standardized CPM and log transform data, you get the basic log fold change and your FDR values. Now, if this was the GLM fit, basically generalized linear model. Now, if you want to do likelihood ratio test, right? So these are like different types of models that are used for differential expression analysis. So basically, if once we have done just the GLM, now we want to check for likelihood ratio test. To do this test, and basically this is your particular p-values and FDR. But you see the FDR is very high. FDR is 1. Now we cannot move forward with FDR1, right? Like we don't want this. So this particular model is not working very well. Now you want to see the top tags. So these are your top, if you just sort that value, log fold change. So you have these particular genes, which are your top genes. Now these are your FDR. Now this is something that we can work with. So FDR less than 0.0005. So that's quite a low FDR. So what it is telling you is the log fold change is associated with a good p-value, which is like 10 to the power minus 7 range. So this is quite good. And you have an FDR low as well. So you have two different tests. So a GLM fit, another is your LRT, your likelihood ratio test. So we have results from this. Now we want to find differentially expressed genes between two separate groups, not like in general differentially expressed. We want to see two contrasts, two different separate groups. Now we don't have a parameter that will explicitly allow to test that hypothesis, so we need to build a contrast. So we build a contrast using make contrast uh, function. So these are your two groups, status pregnant and status virgin, and we have created the design matrix prior here. If you see, so our design matrix has your type and status. So basically type is what is the cell type, luminal basal, and your status is pregnant or virgin or those things. So we have made the design matrix like this. So yeah, yeah, we want to make the contrast. So between two different groups, we want to find a differentially expressed genes. So make contrast is the function, and then we fit a likelihood ratio test. For this one, you can use a GLM as well. So we use the likelihood ratio test, and then we see the top. So basically, these are your top log4j change values, but your FDR is very high. You need to be careful about that. So yeah, so between two different groups, we have got the log4j change values for particular genes, but the FDR level is very high. So you need to be careful about that. 1 second, guys. I'll be back in just a second. But I have the class 7. So what is the thing that you have to see? I'll finish this. Yeah. So sorry for that. Okay. Yeah. So these are your log4 change values, and basically with along with your FDR for two different groups. So these are just like your pregnant versus your virgin group. So basically, between two groups, we are comparing, and according to the design matrix, we are doing that. So this is your design matrix here, right here. Okay. So yeah. So this till this we are getting the differential expression analysis part. Now after this, what we'll do is basically the functional enrichment analysis. So first we started with counts data. We did QC on that. We uh, basically set up, meaning like we did counts per million. We did log2 transformation. We cleaned up the data, Then we checked for differential expression analysis, and then we get different types of models for that. GLM on the LRT, and we check for log-fold change, p-values, and FDRs. Right now, the next part is to get the uh, functional enrichment analysis. Right now, yeah, yeah. So these this is your LRT BSBL is basically your GLM fit for this. So you we save that as a data frame. So the results here. So this is your differentially expressed genes. These are your genes and your log-fold change and other values. So we keep that as a data frame here. Now next, we try to annotate this. So I'll not go into very details of this. So results, and these are your tests. So basically for luminal, you have downregulated of for 3000 genes, upregulated for 1000 genes, and not significant for 11,000 genes. Now we want to add annotations for this. So basically we want to check for the top regulated top log fold change genes, and what are the log fold change IDs, and what are the genes associated for that. Right. So yeah, so we use this particular library that I was talking about in the beginning. So this is kind of a mapping library that you have. So this is a database for mouse. So genome-wide annotation for mouse. We use this to annotate the oncocytes with the yeah. Adjusted p-value can also be calculated using those commands as well. So I've not done the adjusted p-value calculation because the FDR is anyway pretty low. If you look at the FDR, so the FDR is pretty low, so that's why I did not go into that. Not this one, the other one. Uh, results. Yeah. So for the top log fold change, your FDR is pretty low. So that's why we did not check for multiple hypothesis correction, because since your FDR is pretty low, your p-value it's not going to be changing much if you adjust this, right? Because your FDR is pretty low. So if if that is the case, you do not always need that adjusted p-value. But yeah, you can always calculate that using the FDR. So yeah, it's depending on how your data is looking like. So that's how you need to do it. Okay. Now, yeah. So this the package that we are going to use is this one. This for human, it's the same thing except instead of you have HS here, right? So uh, we can use either depending on the sample information that you have. For our case, it's mouse, so we use the mouse thing, right? So we take this library, and these are your columns that we have. So you have different types of ontologies associated with it. So you have your entries IDs from here. You can have enzyme IDs. You can have gene ontology. You can have gene names and ensemble from here as well. So yeah, you can have different types of information that is present in this particular database. So we can use those. So currently we just need the entries IDs mapping. So these are your key types. So we should the entries ID is the one that we are interested in, and this is the key that we are going to use, right? So we use keys, the database, and the key type is the entries ID, and we do that. And so for particular keys, so for these are your entries IDs, you want to check what are your gene names. So these are you give them, like if you give them a subset of keys, it'll give uh, if it is present or not, it'll tell you in that database you can use it or not. Now to annotate, you select this particular database, then you give the keys as your row names, which are your entries IDs, and then you annotate them with entries ID symbol and gene node. So this is how your annotation will look like, your IDs, symbols, and whole gene names. Now, if you want to annotate the results, you do a cbind and check the annotated results. So basically, these are your IDs, and you have annotated them with symbols and gene names, right? So it's pretty simple. Just this is just an annotation of gene names, nothing more. Now we have this whole basically the data frame. So we have calculated the gene expression. We have calculated differential gene expression analysis, and we have gotten the p-values, we have gotten the FDR, and now what we are going to do is basically use functional enrichment analysis, and we'll look into what are the pathways that are applicable to downregulation. Someone is asking something. So LR is basically your likelihood ratio test statistic. It's the statistic for that. The p-value that you are getting, that is coming from the likelihood ratio test, and the statistic that is associated with this, that is given in your LR column. So these are your statistic, and this is your associated p-value. So for the LRT model, you'll get this. This is the likelihood ratio, right? Uh, okay. Now we want to see which pathways are getting upregulated and non-regulated, right? Now we have done a jar and everything. So we'll directly go into FGACA. So this is the package. So FGACA, this is the FGACA package. You can download it using bioconductor only. So, and this is the GitHub page for that. So you can check out the VigNet for this. So we'll do this exact same thing here. So yeah, here is the VigNet for that. And the package is present in the bioconductor. You need to install it, and the information that you need to give it is basically your ranks. So first you need to create the ranks, and then you need to, so basically your ranking of the genes according to differential expression you need to give, and the pathway list, which is basically the set of genes that are present there. So these two things we need to provide to FCACA, right? Now go here. So this is your FGCA library that is loaded. So now we get the ranking of, create the ranking for the particular gene expression, differentially gene expressed genes using log fold change as the parameter. So now if you check results.org, yeah, now it has been sorted using the log fold change values, right? Now you have a ranking of the genes that are associated with it. Now you create the ranks of for the log fold change, and then you check the names, which are the row names, basically the genes associated with it, and these are your ranks. So is this gene, this is the log fold change, this is the gene, this is the log fold change, like this. You have the ranks created. Now, if you want to create the bar plot of the ranks, this is how it looks like. So you have certain genes which are highly regulated, upregulated. You have certain genes which are highly downregulated. Now you want to get the pathways. So the pathways I've got already defined here. So the pathways, if you want to see, so you have for each pathway. So for example, hallmark, allograft, rejection pathway, these are the genes that are involved. So these are your gene IDs. Right? These are your gene IDs. For spermatogenesis, for example, these are your gene IDs. For crash signaling or something, so for each pathway, you have a set of gene IDs. So gene sets, those are, right? So those are the pathways that we are defining. Now we use FGACA We give the pathways, we give the ranks that we have generated from here, and we'll give the number of permutation and the maximum size and the minimum size. Right? Now we run this GSA. Now we check the class of it. It's both our data frames here, and then we check the dimension. So you have 50 and 8. So basically 50 pathways uh, we have done enrichment analysis for. Now we check the results. So for each pathway, you have a p-value, you have an adjusted p-value, you have an enrichment score, and you have a normalized enrichment score, which is basically normalized by the number of gene sets present in that particular pathway, right? So these are your sizes for that. So we have gotten the normalized enrichment and the enrichment scores for each of the pathways that are here, right? Now if you want to plot this, so we can create the enrichment plot. So basically we store the matching pathways and the ranks in a data frame, and then we basically use the ranks and create the bar plot. So basically you have, these are your particular, so the thickness uh, of the lines that are getting showed here is basically the number of pathways that the particular genes is involved in. So it is giving you basically a bar plot according to the rank and the number of pathways that the gene is involved in. But most importantly, we are interested in this. So we are creating enrichment plots. So yeah, see, this is how the enrichment plots look like. So you have enrichment scores here. This is your highest enrichment score, which is reported, and it goes down like this, right? Now, if you want to see which pathway is the most affected here, so we do, like, we check for the top pathways that are affected, top pathways that are upregulated, top pathways that are downregulated, and we like combine both of them up and down, and you can plot the GSA table here. So see, you have hallmark G2M pathway having the lowest p values and the p adjusted values, and accordingly to we have the rest of them. Now if you want to see, for example, uh, if you want to see, for example, uh, one, huh, one second. We use the camera command, which basically takes its input of the DG list object and the index gene sets, the design matrix, and the contrast, and it estimates the correlation for each gene separately. So you can do that for each group. So we do the status and we set the design matrix as the same. Your type and status, basically cell type and your status uh, of the samples. And these are your basically design matrix. So type luminal, status pregnant, and status virgin. And you do a camera run. I think I have not defined C2. Yeah, C2 is here. Okay, I have to download that. I don't have it here, so I'll not go into this detail here. Just wanted to show you how this looks like. So basically, we have done till here, the enrichment scores and the p-values. If you want to see, I'll just show you here in internet. Uh, Pathway enrichment analysis. Yeah, this is how you'll want to present. So you have different kinds of pathways and how enriched it is compared to other pathways. So for example, this glycan degradation is more enriched compared to the other types of pathways. So this is the kind of analysis that we want, along with differential expression analysis. So this is one such plot. This is another one. Sorry, this is another one. You want to check the different types of pathways and what are their log p values changes. So that is how you can say if this pathway is significantly upregulated or downregulated compared to other pathways like that, right? Okay, I think, yeah, so that's all for uh, the tutorial. Now we'll look into the basically the assignment problems for this week. Okay. Okay, the answer for here is set WD because yeah, I showed you in the R command as well. So you just do a set WD here, changes the directory. Now, like say for example, um, just a second. Yeah, I'm using it wrong. So basically, users, yeah, my name, and for example, onwards. Yeah, this is how you change working directories. Yeah, it's correct. The option is B. So basically, the pattern is given by star.csv. Star can be the name of the file. Dot .csv will be the extensions. Yeah. It's the same for Linux commands as well. So if you do ls space star csv, it will list the CSV files in the directory. Yeah, the correct answer is library. So use library in the bracket to give the name of the package. It calls the package. So okay. The answer is A. Basically, if you have given file and from the first column, it's basically your row names is your first column, right? So you have to specify the row names as equal to one, which is your first column. So that way you'll be able to read from the first column. So the correct option is option A. The answer is EDA-seq. I have not uh, covered this, but this is the page for that. Uh, yeah. So within normalization procedures to adjust for DC content level. So yeah, the answer will be A because the variables involved here are your sex, age, and treatment, all three of them. So your design matrix should involve all three of the columns, sex and age and treatment. So the correct answer is A. Okay, moving on. Okay, yeah, this is a multiple option correct. So keep that in mind. Okay, the answer is you need to add the design formula as well. So it takes the design formula, it takes the counts data matrix, and takes the single column where the basically columns or rows of the column data corresponding to the columns of the count data matrix. So yeah, it's correct, but you also need the design formula as well. Yeah, the answer is B. It's basically you're trying to zoom in the x-axis. So just keep the limit between that particular values, 6 and 8. So it'll zoom in the x-axis only. So correct answer is yeah. Okay. Yeah, the answer is RUVC. So we have not discussed this here, but just quickly show. So basically, this uses a mixed generalized linear model to remove unwanted variation from the data. So basically, if you can, this is your original variation, and this is your after you use REBC. So yeah, this is the package that you use to remove unwanted variation from RNA-seq data. We have not discussed it here, but yeah, to yeah, this is the correct answer actually. Okay, the answer is G-plots. It's not feat map. So inside G-plots, there's this function of heat map.to. So G-plots is the package. In our tutorial also we use G-plots for clustering the MDS plotting as well. So G-plots is the package, not feat map. This is a big question. Okay, what the answer that you said is basically okay. Oh, you did not give the answer. So okay, so grep ERCC will work if it is in a Linux or a Unix terminal. So you do a grep, then you give the space and give the pattern that you're looking for and space the name of the file. That will work if you are using a Linux terminal. But if you want to do it in R, so you first need to get the row names, get the basically the row names as a first column. Then inside that you need to do grep of the of this particular uh, pattern in this particular uh, column, basically, which is the row names of the data. So grep ERCC row names, and this will be executed in row name state. So this is how we do it. Normally, this will work. Grep ERCC space your file name will work in a Linux terminal. This will not work in R. Yeah, the answer is B. So this is this package. This is the CAN package, which basically allows you to do clustering based on Poisson model. So that's correct. Okay, uh, the answer is A. So basically, these first three uh, is RGB. So this is red, 100. And now you want to be it little transparent. So add the point 0.3 for the transparency. So that goes up to 1. So 1 will be the least transparent. Towards 0 will be the most transparent. But if you do full transparent, the color will vanish. So you need the color plus you need some sort of transparency, which is given by this point 0.3. So 100.3. Yeah. So yeah, that's all from my side for today. If you have any questions, you might ask now. Acha, PC1 and PC2, it's like basically when you do principal component analysis. So in terms of capturing what you're trying to do is you have multi-dimensional if you have multi-dimensional data set, what you are trying to do is you are trying to uh, get that same type of variation in a single dimension data set. So that is what the principal components mean. So the PC1, PC2, PC3. So the first principal component that will be there will have the highest amount of variance. The next principal component will have a little bit lower, then a little bit lower with PC3, PC4 like that. So what it is meaning is basically the first principal component will be the one that will capture the maximum amount of variance. And as you gradually move down the number of components, the amount of variance captured will be much lower. So uh, I'll just show you one plot here. Yeah, what you're calculating is the eigenvector for that particular data set. So the idea is, yeah, so this is your original data. It's assume there are three-dimensional data. You want to capture the same variation in 3 dimension as given in 2 dimension data. Yeah, sorry. Uh, that is what you're trying to do. You're trying to capture the same variance. So, like basically, what it is happening is, so in PC1, you have the maximum moment of variance. The lower principal components that you go, that has lower amount of variance because that is how it is designed. So in the first species, what you will try to do is capture the maximum amount of variance. So PC1 versus PC2 is a good plot to have to capture most of the variance. So if you do a PC1 versus PC2, what you will see is basically if there are variance between two different data points, they will separate out in the two-dimensional plane. So your original data is multi-dimensional. So what you're trying to do, you're trying to capture the maximum amount of variance in PC1, then a little bit lesser in PC2, then a little bit lesser in PC3. The more PCs you go on adding, the lesser the variance becomes because it is inversely proportional. So the first principal component will have the maximum amount of variance, and the last principal component, which is the so if you have an n-dimensional data set, the nth principal component will have the least amount of variation. So that is how it is designed. So when you're doing principal component analysis, what you're doing is you're creating n different vectors or n different vectors for your n-dimensional data set. So your first vector will have the maximum amount of variance that is captured in the data, and your nth principal component will have the least. So that is how it is designed. So that is why we do this kind of analysis, PC1 versus PC2, to capture the whole n-dimensional variation in a two-dimensional plot. That is the whole idea. Okay. I hope I made it clear. Okay. Okay then. Thank you guys for joining. I'll close the session now and I'll meet you next week. Thank you.